Okay, I think we just get started. It's 1:30. So welcome everyone to this side event where we'll hopefully learn more about the current knowledge we have in the Arctic deep sea, and not least try to identify some knowledge gaps and a way forward to get more knowledge. Today we are so fortunate that we have Margaret Hagen here today to open this event. So she's the rector at the University of Bergen. So welcome.
In the mid-19th century, zoologist Michael Sage retrieved a sea lily from the depth of 800 meters. And with that single discovery, he overturned the prevailing belief of his time could not exist so deep beneath the ocean surface. And today we know more, and yet we still know surprisingly little. The Arctic deep sea remains one of the least explored regions of our planet. At the same time, the Arctic region is under increasing pressure. Ice melt is accelerating at an alarming rate. Commercial interest is growing. Geopolitical interest is growing. Geopolitical tensions in the high north are rising. And when we uncover new arenas, we also uncover new opportunities in the Norwegian Sea. Marine mineral resources have already been identified, and the deep sea is also attracting increasing interest regarding bioprospecting with potential applications in both medicine and production. With new knowledge comes great responsibility. It remains unclear whether the exploitation of deep sea resources can be justified economically, technologically, or environmentally. The University of Bergen was partner of the Go North program and the Legacy After Nansen, and is now a partner in Polhavet 2050, contributing research-based knowledge to policymakers and public authorities. Still more research is needed, supported by long-term, broad-based research programs. Research on the Arctic deep sea has been a priority for us for more than 25 years. This work gained new momentum in 2021 when the Center for Deep Sea Research was continued with the support of the Trond Mohn Foundation. And through the Center of Deep Sea Research, we have built strong interdisciplinary research environment. We have developed advanced infrastructure and made this infrastructure available to a broad international research community. Today, the Center for Deep Sea Research is a key provider of the knowledge base needed for future decisions on the deep sea in the high north. So with this brief introduction, it is a great pleasure for me to give the floor to Associate Professor Stefan Leth Jørgensen, Head of the Center for Deep Sea Research, and he and today's speakers, Owen Reeves, Ida Steen, Mari Eilertsen, and Pedro Ribeiro, will present the current state of knowledge about the Arctic deep sea, and then moderate the panel discussions. Finally, I would very much like to thank you all for joining us here today, and my sincere thanks goes to the panel and to Stefan. Thank you, Stefan.
Thank you. So before you leave the stage, Margaret, this is a small token because we have been supported in our research for 25 years, and it is, as most of you probably know, quite expensive to do. But, and this certainly doesn't weigh up for the millions that we spent, but a small token. Here's a cup from the Center for Deep Sea Research. And thank you for opening this session. Thank you very much. All right. We better get started then. So Welcome, as I already said to you. It's nice to see so many has an interest in the deep sea in the Arctic. Today we will first we will have a number of presenters giving you some updates on the current knowledge that we have about the deep sea, and especially the seafloor and subsea floor in the Arctic. And then we will continue afterwards with a panel discussion, trying to identify some knowledge gaps and how to proceed. There will be no questions immediately after each speaker, so please write down your questions and then we can take them at the very end of this session. Okay, so if we can start the presentation. So because the first thing that will happen is that I will give an overview, very broad lines of what have happened in the last 25 years. So maybe I should start by saying that my name is Steffen Jørgensen, and I'm leading the Center for Deep Sea Research at University of Bergen. I'm also a geomicrobiologist, and my focus is on subsea floor microbial processes. I've been at the university for 25 years, and I've been involved in deep sea research basically from the beginning. So let's see, I need this one, and then do like next one maybe. Here we go. So just a brief rundown of what have happened. So deep sea research started by the current session of deep sea research. I should say, Margaret nicely introduced some of the very early deep sea researchers from Bergen. But in 1999, there was a scientific priority initiative from Bergen that funded a number of researchers from biology and some research from geology, trying to get together and get a better understanding of how geology and biology works together in the deep sea. That was funded through a number of research council projects as well over the next 8 years. But that really made the foundation of what later became the Center of Excellence in Geobiology. And one of the main priorities and objectives within that center was to study Earth-life interactions in the deep sea, with a focus on the Arctic, and specifically the spreading ridges. That center ended in 2017, and then we were lucky enough to get funding from K.G. Jebsen Foundation for a number of years to support our research, and most recently, since 2021, we have been funded by Trond Mohn Foundation. The current iteration of deep sea research and the center today, we have 9 professors, 3 researchers, a number of guest researchers, and Professor 2 positions. So all in all, I think we are 15 permanent staff, and we are currently 5 postdocs and 14 PhDs. And those numbers is basically what we have had over the last 2 decades. We have roughly 20 master students at any given time. So we have produced a lot of researchers and scientists throughout the years with a lot of interdisciplinary knowledge about the deep sea over the years. We span pretty broad in our competence, from marine biology through marine geology, geophysics, paleobiology, geochemistry, and so on. So we have it's one of the strengths, I think, about the research that we have done over the last many years, is that it's really interdisciplinary, which gives that context you really need to understand what's going on in the processes. So, as said, the main funding is Trond Mohn and the University of Bergen, but of course heavily reliant on external funding from the Research Council, from different ministries, from the EU, and so on. Yes. The overarching center objectives in the current center is to lead a national and international effort to explore the northernmost and unknown parts of Earth's spreading system hidden under the ice in the Arctic Ocean, and to deliver this knowledge that we obtain about the deep sea from the polar regions, and deliver that to the public and to policymakers, and obviously the academic community as well. So this is what we're currently doing. The center, as the funding looks now, will end summer 2026, but we are looking into a more permanent and longer funding sources. So going back again and see what have we actually done over these 2 decades or more. we have had from all the time had this focus on geo and bio and the interactions between. And we have focused a lot on the Arctic mid-ocean ridges, looking at seamounts, volcanoes, oceanic crust. We have looked a lot at mineral deposits, how minerals are formed. We looked at development of core complexes. We have looked at sediments, seeps, and hydrothermal systems, not least. And as said, very important to understand that we have been looking at the interaction. So it's not only from a geological perspective or only from a biological, but actually trying to understand how these system works together. Then we also have a strong component in bioprospecting. We will hear more about that later. And then we have brands that are more focused on technology development, and I will talk a little bit more about that as well. So what have we then produced over these 25 years? Quite a lot, actually, if I should say it myself. But so we have led and completed more than 40 research cruises, and we have more than 4,500 data entries in the database that is now online and available. And by data points, I mean you can go into this map now, and you can look at what sort of data do we have, and you can request this data as well. It is all sort of data. It's physical samples, so that we have sediment cores in our repository, we have biological samples in the museum, we have rock collection in the basements. To bathymetric data, seafloor mapping, we have chemical data from pore fluids and from hydrothermal vent systems and from CTDs, of course. We have molecular data in form of genomics from fauna and microbiology. And all that you can go in and you can find this database. There's a QR code here. The database was made available some 4 months ago or something like this. And it's funded by an infrastructure proposal, NORMA, and from the Offshore Directory. Yeah, maybe I don't have to say much more about that, but it's important for us that all this knowledge have now become available to the public. Obviously, we do not necessarily put it out right away. There's an embargo on it so that the researchers that generated the data have time to publish on it. But anyways, it's available there. We have more than 300 dives completed with the ROV, and that translate into more than 100 days of video material, which is significant. But again, this is also, most of this is something that can be requested through the database. The research that we've done is clearly, in many ways, facilitated by a very expensive and say, advanced infrastructure. From the early days, we had GeoSARS, but we have developed both AOV and ROV capability, which is clearly extraordinarily important, very expensive choice, but we have been, as said, helping developing and maintaining these systems, so they are now available also to the broader scientific community. We also are involved in quite a lot of deep sea and seafloor observatories. Just deployed one observatory far up in the north, or in the Frem Strait. And then, of course, we are reliant on biological and chemical laboratories. But one thing I should mention, which is important for the community at large, is how much we've been investing in technology, and specifically in underwater vehicles. From the early days, I don't remember the 1999 iteration of our remotely operated vehicle, but I do remember this one, the Bathysaurus, which was something that the center helped develop with a Bergen Argus company. And back in the days, I have to admit, there was a lot of downtime, and stuff imploded, and it didn't always work. But now we have Egia 6000 that we are maintaining, and as part of the National Infrastructure NORMA, we just got a new funding for that, so we're now in NORMA 2 phase, starting in 2025. And then we very recently, people from the center got money for buying and hosting and maintaining an AUV which will be the first deep water AUV in Norway for doing research. We will have this coming to Bergen in March, I think, and then start the first test dives. Maybe I skip there. Maybe it's a bit of an interest to people that we just installed a permanent control room on Kronprins Haakon so that we can now sit indoor when we operate the ROV, which is a big improvement. I don't need to go into details with these things. So one thing is to collect all these data and have the infrastructure needed, but what do we actually do with this data? Is clearly of vast importance. And I don't have time to go in details with all of it, but just want to mention a few things. We have, when we helped develop the first AUV systems, HUGIN, then we also used it for really, really high resolution seafloor mapping, centimeter scale mapping. And we used that, here you see 2 hydrothermal vent fields, it's this meter scale down here. And based on this high resolution mapping, you can identify different ecosystems, different habitats, and you can use AI now or machine learning to just map autonomous the seafloor and say, okay, this habitat is like this is like that. And this is something we hope to get as part of the new AUV as well. We spend a lot of time on doing video surveillance with the ROV to map biodiversity, again, in the hope that we can use that to get a better understanding of biodiversity on the seafloor and categorize different ecosystems. You'll hear more about that from some of the other speakers. We have done seismic to get a better understanding of the tectonic and seafloor evolution. We have done detailed investigation of core complexes where deep mantle material is brought up to the seafloor, and we try to understand again something about the evolution of along the spreading ridges. How does the deeper mantle look like? What's the composition of these things? This is something that we also have invested quite a lot of time in. We also done a lot of not only seismic from the ship, but also ocean bottom seismometers to understand the finer details about the tectonic activity, how many earthquakes are there, is it equally distributed on each side of the flank system and stuff like this. It's also something we spend quite a lot of time on. Also trying to understand how the fluid flows and how energy and heat is transported from the deeper parts to the seawater. We have done a lot of work trying to understand how the seafloor evolve over time, how fast does it go. There's a recent study by Håvard Stubside who found out that the entire Rift Valley is actually being renewed every 25,000 years, which is quite fast. So new volcanic material is filling up the Rift Valley on that timescale. We are looking into seafloor seeps. This is where we discovered this oil leakage up in the Molloy Deep and sampled that back in 2022. And then, so that's a lot on the, let's say, more geological features that we have been studying. Then we have worked a lot on sedimentary systems. We have more than 400 sediment cores collected along the Mons and the Knipovich Ridge. And we use these sediment samples to look at global element cycles, biological evolution of the deep biosphere, population dynamics, understanding paleoclimate. We try to reconstruct that. There will be a talk a bit later that will go a bit more into detail about that. But you see the map ends up here, so there's not a lot known up in the basins themselves, actually. I'll come back to that. And then finally, this has been a major focus area for the center throughout all the years, hydrothermal systems. And this dates back, I think the first one was in 2005. We found the Jan Mayen vent field, and then just continuing basically every third or fourth year we would find a new system. And the center has been involved in basically finding all the hydrothermal systems with a few exceptions over the year. The most recent one is Ultima Thule in 2023, I think it was, during the Go North expedition. And actually, even more recently, we have this up at the Polarstern mount up here. Just a few months ago, we found something that could be a new vent site up there. But I'm not an expert on the hydrothermal systems, but luckily I have with me someone who is. So Owen, please take the floor and give us a little bit more insight in what's actually going on in these hydrothermal systems and what do we know.
Thanks, Stefan. Thanks, everyone, for a quite well-filled room. I'm going to lead off the talk now with Trio, myself. I'm a hydrothermal fluid geochemist. Ida and Mari are microbiologists and biologists, respectively. I'm going to give an overview of the state of our knowledge of what we know about hydrothermal fluids in the Arctic. And we'll have plenty of time in the panel discussion after about where things are going and what's new. I've been studying hydrothermal systems for 20 years, only the last 10 in Norwegian systems. So let's start with a little bit of background. Systems in the Arctic region form the same way they do anywhere else in the world. The ocean plates pull apart, faults and cracks allow seawater to penetrate, it gets modified and heated up, and then it gets expelled at the seafloor here, which is our hydrothermal vent, forming black smokers, which you may have seen, more interesting cooler vents and plumes, which are more habitable for life, and of course, sulfide deposits. Now that's about where the similarity ends. Everywhere else in the world, or most places in the world, the crust is primarily composed of basaltic rock. It's a bit different in the Arctic. The plates are pulling apart very slowly. You do not have so much volcanism. As Stefan said, it's only erupting every once in a while compared to other parts of the world. So we have infrequent volcanism. The tectonic situation allows you then to start to pull up deeper rock from below the crust, mantle rock, which gives a different flavor to the fluids. And because the mid-ocean ridge in the Arctic has been essentially a glacial margin for as long as time has stood, pretty much since it's been built, you have sediments intermingled in this. So you have many different flavor profiles. I'm a chemist, I'm also a cook, that impart on the fluids here. So let's do a little tour of what things look like. So these various flavor profiles impart one thing: it affects the ability of the fluids to carry metals. And on the other thing, it affects the ability of the fluids to carry very energy-rich chemicals that fuel life. And that's ultimately what's driving the ecosystems down here. So here's the map that Stefan showed again. As Stefan said, the center has been involved in the discovery and or exploration and or sampling of almost all of the hydrothermal vents that you see. I'm personally fortunate enough to have sampled pretty much all of the ones that have been sampled. There are some Pokemon still to catch. And as somebody who studies hydrothermal fluids for a living, I can say that these are the most exciting fluids I've ever worked on. I think this is the region of discovery for new and complex types of fluids. And there's much more work to be done in this area. So we'll start with a little chemical tour. Now the systems to the south around the Jan Mayen area, so Seven Sisters and the Jan Mayen group of fields itself, they generally resemble shallower hydrothermal vents elsewhere in the world. Places like the Mid-Atlantic Ridge or the Mediterranean. These fluids are boiling, they're releasing a lot of carbon dioxide, but that's about as close as things. Interesting, as you move to Mohns Ridge and Knipovich Ridge, one of the first systems that the center discovered was Loki's Castle. Loki's Castle was found to have a very strong flavor profile of sediments. The hydrothermal fluids cooking sediments generates a lot of methane. There are now two other systems that do that: Jotun on the Knipovich Ridge and Ægir farther south. These are among the most methane-rich fluids on the planet. Some of them also have hydrogen, which is also candy for microbes. One of the systems in this area, there's not too many that are inactive, but more and more inactive or extinct deposits are being found. Mons Treasure was for a long time thought to be inactive, but recently activity was actually found there. It has a 38-degree, it's a low-temperature hydrothermal system that is also releasing chemicals to an ecosystem there. So even in areas where we've well studied, there is more to find. Right in the middle of those methane-rich vents is another one, Fovna, which is fantastic. I really love this place. Here, we don't have so much methane, but we have tons and tons of hydrogen and iron. It's by far, at least when we sampled it in 2019, this was the record holder for both hydrogen and iron. I really like this place because the vents are not so hot there, so they're extremely conducive to microbial processes. Microbes don't like it too hot. But they're also really conducive to carbon chemistry and the types of reactions that we're studying for, big questions like the origin of life and prebiotic chemistry. As I said, the hydrogen there was the record holder in 2019. We were all dancing around in my lab, and then essentially as part of the Hawken project funded by the Research Council, we went to Aurora, and then we broke the record again. Yeah, so I'm just going to flip us over the pole now, and we'll look at the Arctic proper, into the Arctic basin. 30 years ago, nobody would have believed that you would have found hydrothermal vents up here. The ridge is the slowest spreading on the planet. There's so little volcanism. But a two-ship operation, German and American, went along the ridge, and all of these red dots are potential hydrothermal sites. The yellow stars are the strongest ones. Only one of these has been tracked to source and sampled. That is the Aurora hydrothermal system here, which one of the projects in the center portfolio was the Hawken project, which was funded by the Research Council. We were fortunate enough to get to this place together with our colleagues here in Tromsø and sample it. People had been trying to get there for 20 years. And we got on the front cover of Oceanography magazine, which is, if you're a scientist like me, it's like Harry Styles on the cover of Vogue. I'm off camera. This is the scale of what we did. Here's Kronprins Haakon, and here's the vent site, and this is 4 kilometers, and this is moving. So the ice was drifting at about 1 kilometer an hour. Yeah. So we had these very, very stressful short dives, bouncing up and down and getting our work done, but we got there, and the very understated peer reviewers of our work said that this was a significant scientific and technological achievement. It was a moon landing for those of us that were there. And so, yeah, this is essentially where we are. We're now sampling places under ice, under moving ice. We actually named some of these vents after other oceans in our solar system. As I said, Aurora broke the hydrogen record. It's going to be a tough one to break because it's enormously high amounts of hydrogen coming out of this system. And we named one of the vents there Enceladus, which is a moon that may have hydrothermal vents rich in hydrogen. Yeah. Also, the University of Bergen was fortunate enough to partner with a Netflix production company founded by the gentleman who had done Blue Planet 2. This was a scientific advisory role, and they were out with us on the expedition. And they basically, we had Barack Obama narrating over this beautiful piece of footage from Aurora. Yeah. So things are getting interesting up there after our sort of moon landing. Last year, we found out that the Chinese had done an expedition on Eastern Gackel Ridge with a manned submersible. So they are, upping the ante. It's very risky to put a manned vehicle under ice. So we have started here. And they are working more in this region. The region, I would say, has never been more accessible, but there's a lot more to discover both on the ridge and south of it. And so with that, I will let Ida jump in and take over with the beasties that live on the fluids.
So Thank you, Owen. And so I will present what we have been doing over the last 20 years in terms of microbiology. I'm a microbiologist and have been involved in the research on microbiology the last 20 years. And the microorganisms, they are actually, I would say, the foundation for life in these environments. So they live on the geofluids that Owen has been talking about, and they can live on, for example, hydrogen, and they can respire as we are doing with oxygen. And based on the energy that they get from this reaction, they produce the carbon that is essential for all the life in these environments. The microbes, they are also very interesting in these environments because we can learn about the limits of life, how far and at how high temperatures can, for instance, microorganisms live. And the record holder is, for now, a microorganism that can live at 122 degrees. But the microorganisms, they are also challenging to study because they are actually invisible to the human eye. So what we rely on when we do our studies actually to study the DNA of the microbes, to learn what they are doing and how the ecosystem is structured. And I can say that when we did our first studies back more than 10 years ago, we thought we did a really good job when we got the DNA of 3 microbes. When we did our study on the Aurora in 2025, we analyzed the DNA of 1,577 microbes. And this is what is also challenging for microbiologists, because in addition to have access to really advanced marine deep sea technology, we have to build these very advanced in-house bioinformatic pipelines. And we know that this will be more and more challenging in the future, but also, of course, they will give us an opportunity to learn, get a deeper knowledge about the systems. So clearly, doing research in the deep sea requires that we have long-term research programs and that we have very strategic investments in what we are doing. And we have studied the vents that were discovered earliest, the Jan Mayen vent fields. They are most extensively studied. Also, the Loki's Castle has been extensively studied, but we are also starting to get some insight into what is going on in the more recently discovered vent fields. And what is important for us is that it takes us from we have the sample in the lab and until we are ready to publish our data, it usually takes 4 years. So this is quite some work, and it takes a lot of time to get the insights and the tools, use the tools that we need to study these systems. And this is my favorite place. This is the Barite field at the Loki's Castle, and I think here you really see how the fluids, the geofluids support growth of microorganisms. So these are actually microorganisms, and inside these tube worms, there are microorganisms that feed them, give them organic carbon. And you also see tiny snails growing on biofilms here, and you see amphipods living on the carbon that the microbes are producing. But studying a hydrothermal vent system is not an easy task. You really need to sample different locations. You cannot just take one sample. You need to know what you're doing. You need to take systematically samples from all the different type of environments that exist within a single vent system. And that is what we have been doing over the years for Loki's Castle. So we have a very good overview of the microbial communities that grow on the black smokers, that grow on in this beautiful barite field. And at Loki's Castle, we also have revealed that there is a subsurface biosphere that actually modifies the fluids. So this is a very good example of microorganisms modifying the fluids and then creating another landscape, another chemical landscape on the surface. So knowing the whole system is very important when you study hydrothermal vents. But hydrothermal vents, they are also really important location to study globally important processes. One of these processes is anaerobic oxidation of methane. So that is a process carried out by two microbes in collaboration, and this is actually the most important process in all marine impacted environments. At Lucas Castle, we have 4 microorganisms that do this job, and they are similar to microorganisms at the Jan Mayen vent field. They are also similar to similar microbes in other vent fields far away in the Guamas Basin, but also in what we call cold methane seeps, which Mari will tell you more about. So this is a globally important process, and Lucas Castle allows us to study this process in detail. Then I will move to the Foven vent field, which Owen introduced you to have a very different chemistry. And this is what we see here. So you see the color of the biofilms growing on these on the chimney surface is very different. So here the microbes actually feed on the high iron that is present in this system. So, yeah, they do not feed on spaghetti, they feed on iron, and they make some insoluble iron components based on their metabolism, and this is what you see. These structures are actually made by microorganisms. But the microorganisms here, they also really want to use the hydrogen that is available in the system. So what is unique for this system is that the microbes that live on iron, they can also live on hydrogen. So this was actually a big discovery for a microbiologist to see this. And it was also of high value for us to have this knowledge when we investigated the microbiology of the Aurora vent field. Because this is also a vent field high in hydrogen and iron. And we actually have seen now, by comparing these systems, that both these systems have microorganisms that live both on iron and hydrogen, and actually they use the same uptake system. So it's of high value for us to be able to investigate more than one hydrothermal vent system, but do this large scale comparison across the whole ridge system. And then I will move to the more applied aspects, because the extremophiles, or the microorganisms that live in these environments, they need to tolerate the high temperature. Their biomolecules need to tolerate the high temperature, and that is a property that has been recognized by the industry for many years. And then, so we have used our in-house pipeline to make a large gene catalog. And we have also been cultivating microorganisms below the seafloor on material that we have received from industrial partners over the years. We have also the ability to actually produce the enzyme and characterize the enzyme. So we have a full pipeline up and running for doing enzyme discovery. And we have discovered enzymes that can degrade alginate. We have discovered enzymes that can degrade cellulase. We have also recently described a novel protease that can work on protein-rich compounds. And all of these substrates, protein, alginate, and cellulose, they are important starting points in production of compounds from industrial side streams. So this is a part of the research that we are doing that give us the opportunity to also find some new solutions for in different industrial sectors. And with that, I will end my talk, and I will give the word to Mari.
Yes, hello everyone. Now we've heard a lot about the hydrothermal vents along the Arctic mid-ocean ridges, and for the rest of the presentation I'm going to expand the scope a little bit to talk about different kinds of what we call chemosynthesis-based ecosystems, because the hydrothermal vents is just one form of ecosystem that's based on this process of primary production based on chemical energy, chemosynthesis that Ida was talking about are doing. And this primary production in the deep sea is very unique because the rest of the deep sea is very food limited because it's depending on, there's no light, so there is no photosynthesis. So it's depending on the primary production sinking down from surface layers. And because it's sinking through a deep water column, there's not so much left of it when it comes down. So there is a general lack of food sources in most of the deep sea. But these habitats are the exception. And in addition to the hydrothermal vents, cold seeps have already been mentioned a bit. These are very often found on the continental margins, the shelves and the slopes. And here you find hydrocarbon-rich fluids seeping out of the seafloor, so very often with methane or even oil. And another type of chemosynthesis-based ecosystem, which is maybe less obvious, are the organic falls. So any large parcel of organic matter, so can be a dead whale or a big tree log, that sinks down to the seafloor, gets broken down by microbes and releases hydrogen sulfide, this process, and generates these slightly anoxic conditions that can again fuel chemosynthesis. And all these ecosystems support specialized communities of animals that can live only there because the conditions are so unique. They're chemically challenging for the organisms, and they have very steep environmental gradients, which leads to specialization. So in order to get access to the food, they have to handle the environment, and this has led to specialization. So here I've expanded a bit on the map that you've been seeing several times, adding also a lot of the cold seeps that have been discovered in recent years. And this is work that's been spearheaded by the University of Tromsø, and through a lot of collaborative projects, I've been able to participate on their cruises and take samples from all these environments. And in addition, I've been trying also to study these organic falls, which is quite challenging because while we can have ways of detecting cold seep or a hydrothermal vent, organic falls, you come across them more rarely and very sort of randomly. But in order to get around this, we have a little cheat. So if you can start, there's a little video here. So we've been doing these colonization experiments where we can basically create our own organic falls by placing pieces of bone and wood on the seafloor, leave them there for at least a year and come back, and they get these colonizations from specialized organisms that thrive in exactly these habitats. So we've been doing this over the last years along the ridge. And the reason why I really want to emphasize that it makes a lot of sense to study these different ecosystems in context is because a lot of the animals that you find here, they are very tightly, both ecologically, but because of some similar conditions, but also evolutionarily linked. So there's a lot of species that belong to the same genera and the same families that have adapted to these ecosystems. And in some cases also, you can find the same species. And then I wanted to zoom out a little bit to explain something about why the Arctic deep sea is so special. And I came across this expression recently that I really like, the Arctic Mediterranean, which might sound like an oxymoron because Mediterranean is usually something when we think about sun and beaches and stuff. But actually what it means is between land. And I really like that this expression highlights the semi-enclosed properties of the Nordic seas and the Central Arctic Ocean, because there are some really strong barriers to dispersal between the Arctic Mediterranean and the surrounding oceans. So the Bering Strait has a maximum depth of 90 meters, which is really shallow. And also to the Atlantic here, we have this Greenland, Iceland, Faroes Ridge, which has a maximum depth of 800 meters. So for the animals that are living below 1,000 meters depth, this is really a barrier to connectivity. And we're seeing this in the fauna that lives in the deep, that there's quite a bit of regional endemism. And this also became very clear when we started studying these chemosynthesis-based ecosystems, because they look really different from what we're used to seeing in other oceans. So in the Pacific, for example, if you go to hydrothermal vents, you will see giant tube worms that can be like 2 meters long. There is nothing giant about the tube worms that live in the Arctic. They are these very thin, hair-like worms, but still, even though they're smaller in dimension, they generate this really rich habitat for other organisms. So inside this, we call them worm forests, there are a lot of little animals that live. And when we started studying this, the Lucas Castle is a good example, we found that there is almost no overlap with other ocean basins on the species level. So the species that you find here, they are really unique, and they are new to science. We have been describing a lot of species from these sites, been giving them names, especially in collaboration with the university museums. And, but there's still a lot of new species, and every time we go out, we find new things that are unique for this region. And Owen and Ida have already been explaining how the hydrothermal vents are very diverse in their environmental conditions in this region. But the same is also the case for the cold seeps. So we have cold seeps from as shallow as 100 meters down to 4,000 meters. And some cold seeps have oil coming out of the seafloor. So when we get up these samples, they are really nasty, smelly, sticky, but still they are full of worms that are apparently perfectly happy to live among this oil. Only some worms, not a lot of different ones. And then we have, let's see if this video, can I try to start the video here? Let's see if it works. We also have these, mud volcanoes that are methane seeps where you have this really strong flux of methane that makes the mud completely fluid, and you get what looks like almost smoke. So the Håkon Mosby mud volcano has been known for a long time. It was discovered in the late 80s. But in the recent years, there's been a lot of new mud volcanoes discovered. The Borealis mud volcano and this Polaris mud volcano province that actually has a lot of different sites. And also in a lot of these seeps, you get these carbonate formations, which is basically rocks that are built from microbial activity, and they generate this hard substrate that really diversifies and impacts the benthic communities. And there's also a lot of fish that really love to live here. And I mentioned depth a little bit already, but this is a very key factor here in structuring the communities in the Arctic CBEs. So this is something that's already quite well known from other parts of the world, and we also saw the same from the hydrothermal vents, that when the shallower vents were explored, there weren't many specialized animals there. But then when we started studying the deeper vents, we saw that this is clearly where the more endemic fauna is found. And the same is the case for the seep. So before, when most of the seeps were known from shallower areas, there was quite a reduced amount of specialized fauna known. But now that we started exploring deeper seeps, we're seeing that there's really a clear specialized fauna there as well. And there's some species that are specialized on seep ecosystems especially, but there's also some shared fauna with hydrothermal vents in similar depths, which is very interesting. And this environmental variability, it probably also influenced the formation of species through time, so the actual speciation process. And this is a hypothesis that we're trying to demonstrate using these tiny gastropods. So these are one of the most abundant animals in vents and also in seeps. You really just get hundreds and hundreds and thousands of them. And to begin with, we only had one species that had a name in this group, And now, after doing a lot of sampling and sequencing, we see that we have at least 5. So these 4 still need to get new names, but we started looking into the evolutionary history and how the habitat might have shaped this group. So we have the species that has a name already. It's actually quite widespread. We find it across both vents and seeps, also organic falls. But you can see from the distribution map here, there's some places we don't find it. And of course, some places that are sort of missing the species is probably because they were very recently discovered and we haven't really studied them largely yet. But some are sort of true absences where we really checked and they're not there. And one case is Fovne, and another case is Aurora. And here, instead, we find this species. And you might remember that this was already highlighted, that these 2 vents have very different geochemical conditions. So here we think that this is a case of specialization to the conditions at exactly these sites. Then we have one species here, which is a bit of a mystery still. We only have quite a few specimens of it, and we don't really understand its distribution yet. But this other species here clearly has a much more shallow distribution and prefers cold seeps instead of vents. While the sister species here has a distinctly deep distribution. So in this case, we think depth differentiation might have been what's been driving the speciation process. So this really shows how the diversity in environments in this area can also drive the diversity in fauna. And another aspect that's very interesting about chemosynthesis-based ecosystems is exactly this that I was mentioning before, the fact that they are so food-rich can also impact the surrounding ecosystems. And one example of this is some of these fish that really love hanging out at the hydrothermal vents and cold seeps. So this is a small eelpout species that we've been studying. And you can even find them sitting on the hydrothermal chimney. On this site, we could measure 100 degrees fluid right next to where the fish was sitting. So it's like they're sitting in a jacuzzi and really warming up. And we have a lot of different hypotheses for why this is. But it's clear that they really love hanging out in the worm forest where there is a lot of food for them. And we're also seeing this in their gut content. They're really feeding on the snails and the amphipods that are super abundant here. And we can also use these stable isotopes to demonstrate that they are getting their food from the specialized vent fauna. So, but they don't exclusively hang out there. They're not an endemic species. They're also seen outside of the vent regions in more inactive areas. So this is a picture from Hausgarten Observatory. And this is one potential process of export of these nutrients from the chemosynthesis-based ecosystems. They might be transporting this out and enriching the surrounding ecosystems as well. So that's another reason why it's important to understand this process. So with that, I can kind of wrap up this little vent trio presentation that we've had by saying that through these last couple of decades of research at the Center for Deep Sea Research, we really built the capacity technologically, but also the systems understanding needed to take the next step and really integrate across this whole region and moving further also into the central Arctic Ocean over the next years. So we are primed and ready to go. So with that, I'm going to pass the word over to Pedro, who's going to talk about some different ecosystems in the deep sea. Thank you.
Thank you, Mari. Wow, it's nice to see such a full room. It's really fantastic. So my name is Pedro Ribeiro. I'm also a seafloor ecologist. There's quite a few of us at the center really focus on. My work is based on non-chemosynthesis-based ecosystems, so other ecosystems and habitats that live beyond active vents or when vents become extinct. So I will just show you a few examples of what we are doing at the center. If I get this right, yeah. this map should be familiar to you already with the Arctic Ridge. I just plotted a few points, a few seamounts that we have visited. The center has visited a lot more seamounts than what are represented there in the scope of mineral resource assessment. The examples that you have here are sites that we consider particularly interesting from a biological point of view, and therefore we are investigating more in depth. And here on this side, you can have a cross-section of the ridge from the deeper part, so that's the axial valley, that's where all the active vents occur, and eventually the seafloor spreads, and the vents will become extinct. But you also have the ridge flanks that, due to tectonics and volcanism, but especially tectonics, can create these fault scarps that we can technically term seamounts. So these are underwater mountains that are at least 1,000 meters tall. And as I will show you further down the road, they are also hosting quite some special life forms. So going a little bit back to active vents, and when active vents actually influence the surrounding habitats and the surrounding fauna, as Mari mentioned, she gave a good example of this. We are also looking at the influence of plumes. You can see on the map down there on the lower left side, the influence of hydrothermal plumes into either excluding background fauna, so that's the fauna that don't like active vents, but also into attracting and creating good conditions for other types of fauna that are quite, I guess this is a laser, that are quite tolerant of the plume. So there's like a sweet spot where we actually find areas of that are densely populated with some species that we understand are adapted to this zone of transition or the sphere of influence of active vents. So we are very interested in studying this area. Of course, the first step is to investigate which species lives there, and then the second step is to understand how they can actually benefit from living in so close to a toxic environment. One of our sources of evidence, again, comes from food web analysis. In this case, is this very non-assuming sponge. So this is a glass sponge. It's actually made of silica, and they can live for a long time. And they are filter feeders, so they take on water, and they filter water, and they take their nutrition from the water. And we have some stable isotope analysis that Mari already showed in her presentation that seems to indicate that at different sites, the same species has different carbon sources. So this may indicate that something that we already suspected, that sponges are quite flexible in what they eat. Let's say they are not fussy eaters, but some of them, particularly in certain areas, depending on how close they are from active vents, they can be getting some food from organic matter that is detached from the vent. And this could be bacteria, for instance. So this is one topic that we are very interested in investigating, is this transition between active and inactive areas in parallel with the dynamics of the ridge. But then eventually vents will become extinct, and we have documented quite a few throughout the years. They become extinct, and then the communities, the vent communities, they are replaced with background fauna. So they are replaced with sponges and other species that are not adapted to active vents. And we have a little example here that shows that on the same site, from active chimneys to inactive sites and to completely background areas, you have a transition of fauna. So this, it's what is showing is that it's not a breakage. It's a continuum, let's just say. And eventually, when the seafloor spreading becomes even further away from the actual valley, we can assume that the vents that we find, or the hydrothermal deposits that we find, are extinct. And in this situation, we find fauna, we find biological communities that are typical of other seamount areas. So we assume that at this point, and for instance, from this active vent here, Aegir, to deep inside, it's around 20 miles. So it takes quite a long time for the seafloor to spread over there. At this point, there is no hydrothermal influence, but of course, we should investigate more closely. And this is the first extinct hydrothermal deposit that has been found, and one area of research should be finding more examples and investigating what lives there. Then going to seamounts. So these are the tallest features on the ridge. This is the Boyd Seamount, is an area that we have investigated quite in detail. And the line, the black line that you see here, is one ROV transect. So it's one transect that we conduct with our underwater vehicle with cameras. So imagine going up, hiking on a 3,000 meter tall mountain and annotating what you see around you. So this is our first approach to understanding what kind of communities are living there. And we see that from the bottom of the seamount to the top, the communities will change. And they will change because of physical factors, because of different adaptability of the fauna. However, these visual surveys should be and are for us the first step to understanding the entire community structure, because we cannot, I mean, we can go quite small. So these are organisms that are probably 1 to 2 centimeters in size, but we also need to investigate not only the microbes, but also the fauna that live within the sediment. So this is work that we are conducting at several areas, and our overall goal is to understand patterns. So by understanding patterns, for instance, what this graph is showing us here is that the communities on the top of the seamount, so there, not only present higher density of fauna, but they also present higher diversity. So by having this complete understanding of what lives on these huge underwater mountains, but it has to be a complete understanding, not just the top, not just the base, we will be able to understand the patterns, and from understanding the patterns, we can investigate the processes that keep these communities there, that allow them to establish and to thrive there, and what is the dynamic of this ecosystem. And for that, we need complementary study methods. So the center has also been deploying underwater platforms. So these are, let's say, either landers or structures with sensors that we bring to the seafloor. They will stay there for as long as the batteries can record data, and then we obtain time series of data. So for instance, temperature, sea bottom temperature, ocean currents, and also biological observations with cameras that we keep down there and taking pictures every few seconds. So it is with this complete approach that we will be able not only to understand, have a good spatial understanding of what kind of habitats we have on the ridge, but also how they live there and what can happen in terms of future disturbance. Can we predict the consequences of disturbance? So just moving forward, I would like to thank, of course, all our collaborators and partners that have been working with us over the years. And just to reinforce the message that in future years, we will have unprecedented access to the deep sea. We can map at centimeter scale resolution. We can obtain sound, so not only optical images, but sound images with centimeter scales. Therefore, we will be ready to, in the next cycle, to go into the central Arctic Ocean and continue exploring and investigating these ecosystems. So with this, I will pass the word to Stefan, who will talk to us about subsea floor biosphere.
I am already out of time before I've started. But I'll take that upon myself. I have not been hard enough on the previous presenters. But Mini, I think it's important to have the panel discussion, obviously. So I will do this a little bit faster than I wanted to. But I'd like to introduce you to subsea floor geobiological processes and what we know about these processes from the Arctic. And from previous experience, I know that I better spend a little bit of time just to introduce what that actually means, because a lot of people are not even aware that one of the largest biospheres on Earth are in the subsea floor sediments. So back in the 70s, based on primarily geochemical data, people started to understand that there is something going on in the subsea floor. There's some sort of transformation of energy. But we didn't really know at that time what was actually driving this. Was it just abiotic processes, with biotic processes, what was going on? Then in the, say, mid-90s, based on some large-scale scientific programs that were drilling deep into the sediments and into the crust, it suddenly became apparent that there was a vast number of microbial cells hidden in the subsea floor. And actually it turned out that approximately 1/3 of all microbial life is in the subsea floor. It's 10 to the 29th microbial cells that's estimated to be there. Put them in one row and there will be 100 million light years into the universe. And remember, they are 1 micron. That's why we call them microorganisms. So there's loads of microbes in the subsea floor. And then in the beginning of the 2000s and the next 10 years, the more we explored, the more we found. And where the first discussions was, okay, so there's microbial cells there, but how active are they? Are they still alive? Are they just relict? Is it just DNA fingerprints that we found? We know now that they are active. We know that they are active because they transform organic carbon. And by transforming this carbon, they are regulating all geochemical fluxes in and out of the seafloor. 70% of the Earth's surface is seafloor. All that's coming in and out, except from the hydrothermal vent fields and the seepage that we just heard about. It's microbes that are controlling this. Greenhouse gases in and out, oxygen in and out of the seafloor. Okay, we also know that the microbes that we find down there are very different from what we see anywhere else. So there's a very high diversity, and they are uncharacterized. Basically, when we started to investigate these systems, the tree of life got suddenly almost double the size. Okay, so that's just background. Let's move on. What do we know about it in the Arctic? So the initial studies were primarily done in the Pacific Ocean. We started to investigate the Arctic in around 2007, 2008, and collected some of the first sediment cores where we have microbial data and geochemical data together. And then we have kept investigating that over the last 20 years or so. It's important to notice that we have a great focus on recovering a lot of data, not only looking at microbial diversity and find out who's there and characterizing them, but actually to understand what they're doing, we need context data. We need geochemical data, we need physical data. So here's just an example of what we do with many of our sediment cores. We map it in terms of isotope data, grain size. We look at what sort of solids are in the cores. We look at the lithology. We look at the pore fluid, what's in the chemistry in the pore fluid, what's going in and out of these systems. And then we look at the microbial biology in terms of looking at how many are they, what are they doing, what are their function, how are the community put together. And it's highly complex systems. In 1 gram of sediment, you find approximately 100 million to 1 billion microbial cells with a high diversity. So it's very complex systems. But luckily, they are also very stable, at least we believe. Then we take all this data and then we transform it into more dynamic data. So we use the chemical data to calculate fluxes in and out of the seafloor. We look at the paleoclimate record. When was this sediment deposit? What sort of temperatures were there then? How much organic carbon was deposited? And then we transform the pore fluid data, for instance, into reaction rate. So we know, okay, the microbes are performing reactions at this and this rate. We know how many microorganisms are there. So we know how much energy they need. All those sort of things is something that we look into. Yeah. I have absolutely no time to go into details with everything we've done. I'm just going to take a few examples of some of the studies that has been performed over the years. I think one of the first one that we did is a publication where we link microbial community profiles with geochemical data. And that's actually, I think this was published in 20, something like that. There was the first study to actually link that the microbial composition is determined or very highly correlated with geochemical data. This was the first study to actually prove there's a significant correlation, and that tells us something about how the communities are put together and why they look the way that they do. We also have another study where we investigate how to what extent are they active? To what extent do they actually grow? So the microbes fall down on the seafloor and then they start to eat organic carbon, but then they get buried. And for long it has been discussion about, well, when they are then covered by new sediments buried further and further down, what happens? Are they still metabolizing? Are they still active? Or are they just in a dormant state? And we were able to prove that they are probably in a very low metabolic state, and then they hit these transition zones where there's chemical energy, and they start to grow again. And they can be buried for hundreds of thousands of years and then getting reactivated and start to grow. Then we have done some study looking at what are they actually, what sort of microbes are there. And one, just going to highlight two studies. One, a really important study that was published 10 years ago now actually, where we found an entire new group of archaeal organisms, and describing that and characterizing that showed us that this is a archaea that's one of our most recent microbial ancestors. So it sort of was a paradigm change in the way that we look at evolutionary biology. It was also a study that's now changed the whole topography of the tree of life. Then curiosity is this study that actually got a lot of attention. Marine sediments illuminate chlamydia diversity and evolution. Before this study, I can't remember, maybe this was 2018 or 19 that we published that. And I thought it was just going to be another publication that didn't really had any attention. But because it was chlamydia, the media really liked it. And before that time, chlamydia was not known to be in marine sediments. And the reason for that is that the method that we have been using completely overlooked this group. But now we know that chlamydia is very, very abundant in marine sediments. And it's likely to be there without a host. And all sort of things that makes it very interesting to study, actually. These are just some examples. There are many more that we have had over the years, but as you can see, they are primarily data and samples that we have from the ridge system, and not so much from the flanks. So there we could do some more. Just to wrap up on what we know, this is, I mean, I don't know how interested that is, but we know something about the organic carbon content. We know something about the fluxes in and out of the seafloor. We know something about how many microbial cells that are there. We know something about diversity, but we also know that there is still a lot of this diversity that we haven't really characterized. We have a name, but we don't really know what they're doing. We know how much energy they are consuming. We know what they're feeding on. So we know quite a lot, I'll say, but there's still a lot that we don't know. One of the things I just want to highlight before we finish here is that here's some of the things that we don't know. This is the Central Arctic Ocean. These are samples which is more than 0.5 meter deep, where we have microbial and geochemical data. And it's basically nothing. So we don't know anything about what's going on in here. Obviously, we can assume that the functions of these ecosystems are more or less the same as we know from other places, but exactly how they work, we don't know. And especially, we have no idea how they would respond to perturbation. How will this change when the Arctic Ocean starts to change, temperature increase, the sea ice is melting, organic carbon fluxes will change. So this is one of the big things that I think we still need to investigate. And with that, I did actually spend the time as I thought. Okay, anyways, we are in a hurry. So I will skip this, and then I think we can move on to the discussion part of it, where we'll try to identify some of the knowledge gaps that we know are there, and how we potentially can close them. So I'll start by just having the speakers up here. If we can put take the chairs up here, and then we have a little bit, just take a chair up here. Yeah, if you want to stand, then please stand. I mean, because we don't have an awful lot of time, so you won't be standing for a long time. But okay, we have everything. So I'll start by asking a few questions to the panel, and then we will have open for some questions afterwards. And since we are a little bit short on time, I really urge you to keep it short. But my first question would be, within each of your respective research fields, what would be the most pressing knowledge that we need to gain? What do we not know, and what do we really need to know, in a few sentences? I'll start by you, Owen.
I would say diversity of flavors, if you will, of Arctic fluids. I don't think we've really understood the breadth of that spectrum. Every time we find a new place, we find a new composition, so the goalposts keep moving. And so there's a lot of discovery to be done, and then follow-up work after that.
Right, so two follow-up on that would be, why do we need the full flavor?
As George Mallory said, because it's there. We live on this planet, and it's the equivalent of not going out in your back garden because it's raining, or maybe there's some spiders or something. The Arctic Ocean is there. It's Earth's last unexplored frontier, and we can either choose to explore it, or we can leave it.
And then the obvious next question would be, how do we close this knowledge gap?
I think this requires, finding a hydrothermal vent is a complex process. Sampling it's a complex process. We've gotten very good at that now. Once you take the samples, the work really begins. It requires a long-term strategic investment. It's not enough to get one cruise and one trip or one paper. It's always going deeper. So it's sort of it's a strategic level investment, I would say. We've been doing it for years, and we've gotten very good at it. But there is an unlimited amount of stuff still to do.
Thanks, Owen. And I can pose the same question to you, Ida. So what's the most pressing knowledge gap that you would like to close?
Yeah, so when we have seen the diversity of in type of systems that exist already in the locations that we have studied, and with the knowledge about that they will be even more diverse further north, it's obvious that we need to also know the microbial diversity and what they are doing there. But there is also one thing that is really important to investigate, and that is to investigate how related the microorganisms are that colonize these different environments, and also similar type of environments, or environments like cold methane seeps, where there is the same menu that we see for the microbes as we see in the vent systems.
And to close it, you need?
So, of course, we need to do a lot of sampling, and we need to, or new locations, but we also need to be very systematic in the way we analyze the samples, and we need also to develop and be ready to analyze really big data sets. So I would say for microbiology, that is a big challenge, but also a big opportunity.
And Mari, if I've asked you the question, then what would be the knowledge gap that you need to close?
I would say that we still have a long way to go in understanding which organisms we find where, so their distribution and their habitat association, which is really important to understand their vulnerability also to changes, whether that be man-made or natural variations. And then also to move beyond just characterization to really understanding the processes and the drivers behind the communities that we're seeing. So really linking them to the environment and how that's shaping the communities.
And again, for you, what do you need to be able to close that gap?
I mean, we always need, no, not always, but we do need additional samples. But I would say that there's a big gap to fill in what happens after you come home from the cruise, and actually having skilled, trained people to sit and work with the samples to identify them, sequence them, which also generates data that will be really important for future monitoring efforts. If we want to use eDNA, if we want to use video surveys with machine learning processes, we need the data to ground truth these methods and really say what are the biases, what are we capturing, and what are we missing.
Thanks. And Pedro, same question for you.
I think the current knowledge is quite fragmentary. Part of it because some of these habitats are actually isolated, so they are fragmented by nature. But knowledge is fragmented. I think we need a holistic understanding, not just of distribution, but also of ecosystem functioning, to be able to have a fundamental understanding of how the deep sea works, how the Arctic deep sea works, but also to establish area-based management tools to face future challenges.
So to get a holistic understanding, what do you need?
Well, I think we need more resources as they are coming. Of course, we know it's a continued effort, but we also need to bridge, as Mario was saying, bridge this link between obtaining the basis of knowledge and transpose it into a technology that can actually help us analyze the data more quickly. So not only analyze, but so we need technology to collect the data efficiently, to store it efficiently, to share it, and to analyze it efficiently, so that then we can develop methods that will take some of that burden from humans, and we can use machines to at least give us some patterns that we can then analyze.
Okay, so more robots. And some good humans to supervise, I suppose. Yeah. Okay, and with that, I think we can have a few questions. Is there any questions from the audience? And I shall walk around with this one. Start from one end, yeah?
Achim Randelhoff, Aquaplan Neva. Thank you. It's a very, very nice overview. They all flowed into each other. The presentations was very good. My question is based on species distribution and genetics and those things. What can we know or hypothesize about dispersion between those ecosystems?
Oh, yes, oh, that's me. Who wants to take that?
I can say something, yeah. I would say that this is something we're still in the early days of exploring, and there's a very strong correlation between the inherent biology of species and the way that they are dispersing and maintaining connectivity, and this is also shown in their distribution, so how broadly are they able to be distributed. But a challenge with this is that even though we're starting to do these kind of studies on some taxa, it requires us to sample very broadly to find general patterns that can then be used in modeling and so on. Yeah. Anything to add? No? Good. Yeah.
Thank you. Hello, Karin Buhmann, Copenhagen Business School. Thank you for an excellent presentation that actually made it clear to somebody who with a social science background what much of this is about, and these are very dynamic processes once we look below the ocean surface and to the bottom. My question is on the general research that you do, taking into account that Norway has announced policies, currently paused, but for deep sea mining. How do you see the research that you do in any way sort of informing the decision making in regard to what may come of those policies?
I think I can follow up maybe, but you can start, Peter. You.
Thank you for the question. I think the center has been one of the main providers of information and scientific data during the opening process, in this case for seabed minerals. So we assume that responsibility based on 25 years of research, but also having a good overview of the areas where these minerals occur. However, we were also always very clear that there are knowledge gaps that impede a solid, let's say, impede a solid management strategy. So we need more information, we need more science to provide robust answers that can be translated into management. And of course, we are...
Does it just relate to the minerals or also to impacts?
Also to impacts, of course.
What is the time scale on impacts that you look at? Is it sort of, you say, how many years?
Yeah, are you referring to seabed minerals in particular?
For mining of seabed minerals, what would be, say, the environmental and ecosystemic about really rich ecosystemic activities, right? So, could you say a little bit about the time frame of the impacts that you work with, or that you
Yeah, so there are two types of impacts, right? There's the direct impact, which is immediate, and then there's indirect impacts that are mainly caused by the spread of plume. So we have not, there is no conclusive studies whatsoever right now regarding the spread of plumes. So I'm not the best, I'm not a physical oceanographer, so I think that's one good example of a study that needs to be conducted in order to understand how far plumes can spread, if they spread at all farther away from the mining site, and what are the long-term consequences of this spreading. So there are two time scales, right? The immediate and then something that can take longer. I don't know.
No, I think that covers it quite well. I think, yeah, just to add a little bit, one thing that I see where the center, as Peter said, the center has already, we are given all our information further on in the, to the government, so they have the baseline. What do we know now? What we clearly don't know is what is the long-term effects of this? How vulnerable are the different species that are there? But I think it's also important to say that if we can understand the ecosystem function in these areas that might be impacted, then at least we can say something about, okay, let's assume worst case and you destroy all the diversity here. What does that actually mean? Except that obviously you remove some diversity, but is that critical in the sense it's the same diversity somewhere else? Does that really matter? And what is the function of that particular area? Is that something that will have a local impact or global impact? And this is also let's say studies and investigations that we are looking into and hopefully we'll be able to provide. But it's all a matter of resources, isn't it? How much can we do and how much information can we produce? So, yeah. And time. Clearly, yeah. More questions? There are 1 minute and 30 seconds left. No more. To wrap up, and I'll just say something then based on what I think is going to be. I mean, so you all, the panel now said, okay, what do they need? And I think a lot, as you can see, a lot of it goes, and we need to investigate even more. Like, and especially the way I see it, with the place where we really need to investigate now is we don't know anything about the central Arctic Ocean. So to have this dedicated program, Arctic Ocean 2050, is hopefully really going to help our understanding of that, because we know so little about what's going on up there. So that's going to be exciting, and I think this is where we really should push the next decades. Yeah. And with that, I'll wrap up, and thank you all for coming. Yeah.